In principle, anywhere where you see high throughput biology, in principle, we can do it better. That's a little bit of a generalization because it comes down to things like chemistry, which are really difficult, and importing chemistries from one platform to another sometimes doesn't work actually. But in our case, we have products in the pipeline that will do very high throughput, for example, genomic screening, protein-protein interactions, protein-ligand interactions, all things that are cutting edge and very important. And we've also been able to do the very beginnings of what could be some fundamental science. If you think about our bodies and about how biology is done at our level, it's actually done in a microfluidic environment. When you boil all the way down to a cell, for example, it's plumbed by very tiny capillaries and things that take away the waste. It's all done with very, very tiny plumbing. And we've done some early experiments that are at least suggestive that when you create environments on a chip that allow cells to grow and then combine that with plumbing around them, things to bring in oxygenated fluid and take away the waste and stuff like that, that we may actually be tricking the cells into thinking that they're still inside the body. That's a fundamental result if it turns out to be true. If it turns out that we're able to trick cells into thinking they're still in vivo, then there's all kinds of cell biology that you could do that is impossible to do today. So those are the kinds of things that we're working on. They range from very high throughput methodologies to new science.